and good afternoon, everyone. And I'd just like to thank the organisers for the very kind invitation. It's a real pleasure to be here at such a great meeting in Edinburgh. Just by way of a very quick overview, I've been asked to talk about the B-cell receptor. So I'm going to give a brief introduction to some of the key aspects, but I'd really again like to thank my two preceding speakers who've made that job very easy for me. But I did want to share with you some new data, and so I'll move on to an unpublished study and tell you about some of our results looking at targeted inhibition of B-cell receptor signaling, particularly its impact on mRNA translation and how we might be able to tease out differences in the response of CLL and normal B cells. So we heard quite a lot about the B-cell receptor, but I really want to stress 2 points that perhaps we don't talk quite enough about. There are two main functions of the B-cell receptor. So clearly one of those is signaling, and we tend to focus on the signaling capacity of the B-cell receptor because it's obviously so important in terms of drug responses. But the other main function of the receptor is antigen internalization. So following antigen binding, the receptor will internalize, that antigen will get processed and then presented. And that's absolutely critical for normal B-cell responses, probably as critical as signaling, because it's important to gain T-cell help, and without T-cell help, there won't be an antibody response. Also, in terms of the signaling responses, the receptor is a very dynamic molecule. So certainly it can, in certain contexts, it will drive signaling pathways leading to proliferation, survival, and activation, particularly in the context of T-cell help. But in other contexts, it can drive pathways which hold B cells in check. So antigen engagement can, in some situations, drive B-cell apoptosis, and something that's particularly, I think, relevant for CLL, it can drive B cells into this state called anergy. And this is sort of a very intriguing sort of state that B cells enter. They remain viable, but they're lethargic. They're metabolically compromised, and they tend to lose responsiveness to signaling through cell surface receptors. Now, I've been asked to say a few words about CLL-specific features of the B-cell receptor, and many differences between normal B cells and CLL cells have been described, but I think the most striking one is actually if you simply look at the expression of the B-cell receptor itself on the surface. And in fact, CLL cells have got a very low level expression of the B-cell receptor compared to normal B cells. And that receptor, in many assays, seems to be quite functionally compromised. So the levels of signaling that it can induce is often very low. And that may seem like a surprise. So we often think about CLL as a B-cell-driven disease. And that's clearly illustrated by the very dramatic response to inhibitors targeted against these signaling pathways. So there's a conundrum here. We have a tumor which seems to be dependent upon these signaling pathways from the BCR, but if we actually look at the BCR itself, it's there at low levels and is functionally compromised. And I've drawn here on your right a model that sort of reveals what we think might be going on. So the most prominent response to antigen engagement in CLL, we think, is actually induction of anergy. This strange sort of cell phenotype, or the cell state of lethargy. And that certainly explains the very strong downregulation of surface IgM on CLL cells. But anergy is reversible, so cells can recover from this state and likely undergo repeated rounds of antigen stimulation. And it's a balance probably between anergy and the positive signaling from the B-cell receptor that determines outcome. So where anergy predominates, that's associated with indolent disease, classically mutated CLL, but where there is also positive signaling, that may just tip the balance towards the accumulation of the clone and activation of growth, proliferation, promoting pathways. And positive signaling seems to be more common or more observed in the progressive subsets, and particularly unmutated CLL. So I think the key question I just want to talk about is any of this function of the B-cell receptor really specific for CLL? And my view is probably it isn't. I think really what's happening is that CLL cells are obeying the rules that normal B cells obey. And that's really what we're seeing. So induction of anergy, induction of proliferation, they're perfectly normal B cell responses under defined situations. Is there a role of genetics in the dysregulating BCR signaling pathways? Well, a number of different mutations have been described that might impact on BCR signaling and presumably in the patients where they occur, they may be clinically important. But in general, they're present at a low frequency, and they tend to impact on just a single arm of the BCR signaling response. And I think overall, there probably isn't strong evidence that there is a profound effect of genetic change on the function of the B-cell receptor, certainly not as a common hallmark of CLL. I think there is a very important impact of genomics, and that is to do with BCL2. As you know, BCL2 is activated in many CLLs because of the 13q deletion. Now, normal allergic cells have got a short lifespan, but I think BCL2 is maintaining the survival of CLL cells and just opening a window in which they can undergo this sort of recovery and re-stimulation. So it allows a window in which they can undergo these repeated stimulations. And just very briefly, I think we really need to think carefully about what normal cells we compare to in our studies, and this has been mentioned a couple of times already. So when we look at CLLs and compare the normal blood CD19 cells, what we're really doing is comparing a clonal B-cell population to an extremely heterogeneous mix of B-cells and polyclonal B-cells. So they comprise naive cells, memory cells, switch cells, rarer B-reg, allergic cells. So it's really no surprise that we can very often show a difference between a CLL cell and normal B-cells. But we probably need to look much more closely at the normal B-cell subpopulations to determine if they're really distinct features of CLL cells. And so I want to switch my attention to really the positive signaling downstream of the B-cell receptor and potential drug targeting. And you'll all be very familiar with diagrams like this and a lot of interest in the kinase inhibitors, but clearly one of the emerging problems is resistance. I want to talk about a distinct strategy to potentially interfere with these pathways, and that is interfering with their downstream effects. And many of these pathways feed into changes in gene expression by regulating transcription and translation. And whether we can interfere with translation as a way of depriving the tumor cells of this upstream signaling capacity. So I'm going to talk about mRNA translation, which was interestingly just mentioned in the previous talk but one. For many years, mRNA translation was looked at as a housekeeping function, but we now know it's incredibly sophisticated and tightly regulated. A lot of that regulation happens at the level of initiation translation and through this complex 4G, which comprises these initiation factor proteins. 4E is particularly important because it binds the M7G cap on RNA, and that's important for bringing in the ribosome to start protein production. And it turns out that some RNAs are particularly sensitive to the levels and activity of these complexes. And an important oncoprotein is MYC, which is very sensitive to the function of this complex. We tend to think about MYC in the context of higher grade lymphomas, but I think it probably is also important for proliferation in lymphoma. And certainly its expression is induced following stimulation of the B-cell receptor on CLL cells. And we can also demonstrate expression of MYC in a fraction of the cells, CLL cells in proliferation centers. And so the idea is that because of this specificity, we may be able to use drugs, if we can be sufficiently refined in our approaches, to interfere with the activity of these components, and that might allow us to selectively inhibit the expression of certain oncoproteins. So our interest started when we were looking at activation of translation in CLL, and we compared global mRNA translation, activation by anti-IgM in both normal B cells and CLL. And you can see here that we actually stimulate translation in both of those cell types. That's not a great surprise. But what was interesting is that when we looked at the components of that initiation complex in CLL, we saw a very profound restructuring. So there's an upregulation of 4G, upregulation of 4A, and a downregulation of PDCD4, which is a natural inhibitor of that complex. By contrast, in normal B cells, we saw none of this regulation. So this regulation is specific for the CLL cells. So what do I think is going on? Well, actually, I think that CLL cells have got a low translational capacity compared to normal B cells. So they need to increase their translational capacity following stimulation to support translation. And it probably isn't specific for the initiation. We've also described differences in the ribosome components of CLL cells and normal cells. And again, it's surprising, it's the tumor cells that have got the lowest levels of the ribosomes, not the normal cells. But that opens up the opportunity that we may be able to selectively interfere with translation as a way of selectively interfere with these initiation factors as a way of blocking translation in CLL cells. So we focused on EIF4E, which, as I mentioned, is a cap binding protein. It's critical for translation of some RNAs. Interestingly, it also plays a role in nuclear export of RNAs, again, just a subset of RNAs, and that's via its binding to a protein called XPO1, which is interesting in CLL for other reasons. We've used this drug called ribavirin. Some of you will know this. It's an antiviral. It's a nucleoside, but actually you can see it's quite a close structural mimic of the M7 cap, which is what EIF4E binds to on RNA. And it's some very nice work from Catherine Borden showing that the ribavirin will bind to EIF4E and it will displace the M7 cap and release RNAs. And so ribavirin we expected to block both of these functions of EIF4E. So this is the effect on global translation, and we're using a FACS-based assay to measure translation, which allows us to gate and look at translation in specific cell populations. So this is in the CD5, CD19 cells in a CLL PBMCs, low levels of translation without stimulation, and then we see this nice induction of translation following activation of the B-cell receptor, and that is inhibited in cells treated with ribavirin. And 10 micromolar gives us about a third of a reduction in translation. So it's a partial effect, but that's really what we expect because not all RNAs are going to be dependent upon EIF4E to the same extent. In normal B cells, again, we see a nice induction of translation with anti-IgM, but this is essentially unaffected by ribavirin. So it looks like the ribavirin selectively will decrease translation in CLL cells, but not these normal cells. We've looked at MYC expression as well. So here's MYC protein expression. Again, the protein is induced by anti-IgM, and it's nicely countered by addition of ribavirin. And actually, we've probably reduced MYC by a greater extent, 70%, and we also see effects at lower concentrations of ribavirin. We think so MYC looks particularly sensitive to this sort of inhibitory strategy. It's selective, so there's other proteins such as MCL1, which are unaffected by ribavirin. So here's the induction of MCL1, not affected by ribavirin, and in fact, we see very little apoptosis in cells treated with ribavirin even out to 3 or 4 days. In terms of mechanism, we've looked closely at the RNA for MYC. This is total RNA. Here, you see this increase in MYC RNA in these cells, but that transcriptional response is not affected by ribavirin. Where we do see a clear difference, and this is how we think ribavirin is working, is by affecting the distribution of MYC RNA between the nucleus and the cytoplasm. So it's affecting its export from the nucleus. So we've separately prepared cytoplasmic and nuclear RNA, and we're looking at the distribution. Following anti-IgM stimulation, we see an accumulation of MYC RNA in the cytoplasm, where it's available for translation, and that ribavirin completely reverses this cytoplasmic redistribution. And there's several lines of evidence, actually, it looks as if the real role of EIF4E and MYC control here is effect on nuclear export rather than translation per se. And then finally, in terms of data, I just want to show you some results, some in vivo results with ribavirin. So these are TCL1 model, it's adoptive transfer of leukemic cells into mice. Ribavirin we're dosing here at 80 mg per kg, and it's quite easy to dose ribavirin, and we don't see any evidence for toxicity in these experiments. The blue line shows the accumulation of leukemia in the control animals. There's a sort of rapidly progressing leukemia in this model, and here's the results of ribavirin. And so we see a substantial reduction in the accumulation of leukemia in these animals. Now, bear in mind that ribavirin wasn't particularly pro-apoptotic in our hands, and we think it's primarily acting as a cytostatic agent. So we also combined ribavirin with venetoclax. So the green line is venetoclax alone. Again, it's partially active in this model, but the combined effect of ribavirin with venetoclax gives us really quite strong tumor control in this model. So in summary, I think we probably all agree that BCR signaling is absolutely a key determinant of the behavior of CLL cells. It's clearly a very effective target for drug therapy. But I think that the behavior of CLL cells probably largely reflects the normal influences of antigen on B cells. Now, there may be a role for genetics in individual patients in changing that response, but I don't think epigenetics, I don't think the BCR signaling is globally changed by the genetic makeup of CLL cells. And in terms of translation, it may be that differences in the way that translation is regulated between normal B cells and CLL cells might provide us some novel therapeutic opportunities. By targeting signaling down at the bottom of the cascade, it may be a way in which we might treat patients which have accumulated mutations or signaling rewiring in their upstream pathways. And ribavirin, so it reduces anti-IgM global translation in CLL cells, but not normal B cells. And I've just put normal in, just sort of flagged it up to remind me, again, come back to my caveat, it really matters what normal B cells you compare to. So we're comparing to CD19 naive memory B cells. I suspect if we went in and looked at normal human anergic B cells, we might see some very similar properties, okay? So it probably reflects an anergy again in these cells. Ribavirin seems to be predominantly cytotoxic. It prevents accumulation of MYC, and I haven't shown it, but also cyclin D1. And both of these seem to be driven by an effect on the nuclear export dependent upon EIF4E, and then ribavirin is effective in this TCL1 mouse model alone, or nicely in combination with venetoclax. So I'd just like to thank my co-workers. I'm not going to go through them all, but I particularly just want to acknowledge the contribution of Sarah, who's just finished her PhD and did most of the in vitro studies on EIF4A and EIF4E inhibitors, and also the main funders for the work. Thank you.
Thank you, Graham, for these exciting new data. We have time for 2 questions. Please.
Thank you very much for your presentation. Our group and the group from Frida Stevenson, we demonstrated many years ago that the low expression of the receptor in CLL is the consequence of a defect in the assembly of the receptor, and as a consequence, it was retained in the reticulum endoplasmic. And it was also a defect in glycosylation. Almost the great majority of CLL cases respond poorly to stimulation, and Dr. Marti showed the initial work from Nick Urasi demonstrated that there was very poor response. Now, when you stimulate and you get some response, could you see what happened with the assembly of the receptor? Is this going to correct, which in that case could be the consequence of anergy?
Yeah, I think you're right. We've in Southampton obviously worked very closely with Frida and been pivotal in these studies. I think one of the important things is if we, you're familiar that if we leave these cells in culture, they naturally recover surface expression. So I imagine that they're naturally reversing some of the effects that you've described. So I think it's occurring naturally in the culture. I don't think it's driven itself by BCR engagement. If anything, that's going to re-internalize the receptor.
Maybe one more question from me. What do you think at this time is the most physiologic way to stimulate the BCR in vitro? What do you use when you say anti-IgM?
Yeah, I mean, that's an absolutely great question, and I don't honestly know what it would be. To reflect what's going on in vivo, probably soluble antibodies aren't the right answer, and probably in vivo, we're talking about engagement of immobilized antigen, and it likely has quite different effects from a soluble. We're using a bead-bound form of anti-IgM, which does potentiate the responses, and perhaps it mimics some of that immobilized response a little bit more. But I think, we're looking at models which aren't fully going to recapitulate what's happening in vivo. I'll be the first to accept.
And IgM versus IgD stimulation?
Yeah, IgD is different and not fully understood. So most CLL cells retain the ability to signal through IgD. And it's not it doesn't recover in the same way IgM does. But IgD, I don't think anyone really understands what IgD does in normal B cells. In CLL, many samples can signal through IgD, but the responses seem to be very short-lived, and they don't engage these downstream effector mechanisms. So IgD is a very poor inducer of translation, actually.
Thank you.
Thank you.
Thank you again.